Good morning. My name is Rowan Tinker, and I'm a sophomore at Eastside High School, and this is my project, Bacteria Biofuel. This year with my research project, I wanted to focus on the genetic modification of a bacteria for ethanol production. Right now, ethanol production is inefficient and expensive because not all sugars available are being fermented in the fermentation to produce alcohol. There are two main types of sugars that need to be fermented to produce alcohol, and these are hexose and pentose sugars. Right now, not all yeast and bacteria, our fermenters, have the ability to ferment pentose sugars, but they can ferment most hexose sugars. So I wanted to produce a modified bacteria that would be able to ferment both hexose and pentose sugars to improve ethanol production. So in my project, I took bacterial DNA from E. coli and was able to successfully extract this and put it into this new bacteria called Pediococcus pentosaceus. After the new DNA was transformed, I would be able to have a bacteria that could ferment both hexose and pentose sugars. So in my project, I went to Trine University to complete the laboratory parts of this genetics experiment. I started off with DNA that was housed in E. coli in what is known as a plasmid, and I was able to successfully extract this plasmid from the E. coli. I grew my E. coli in an incubator and was able to extract the DNA using a plasmid extraction kit. After I was able to extract the DNA, I needed to make sure that I successfully extracted it from the E. coli cells. And after running a nanodrop and electrophoresis test, I was able to conclude that I extracted my DNA correctly. After this extraction, I needed to get my new bacteria into my Pediococcus bacteria, and I did this using heat shock. Heat shock uses a 47 degrees Celsius water bath, and it loosens up the cell walls of my Pediococcus bacteria to make sure the new DNA is inserted into the cell. After concluding heat shock, I needed to test to make sure that my bacteria was transformed successfully. I did this using antibiotic resistance. Only bacteria with the new DNA would be able to be grown in the presence of an antibiotic called ampicillin. This is my plated bacteria in an ampicillin plate. Since it was able to grow in the area of ampicillin, I made sure that I transformed the DNA successfully. After my new bacteria was grown, I had to produce ethanol from it. To make sure that I had a control sample, I produced ethanol from my Pediococcus bacteria with samples that had the new DNA and samples that did not. After I produced this ethanol, I had to test to see how much alcohol content did I produce. And I did this using gas chromatography. I used a gas chromatograph to test my alcohol content of my samples. So I tested 3 different samples. A sample of a known 1% alcohol, a sample of my new bacteria ethanol that had the new DNA, and a sample of my new bacteria ethanol that did not have the new DNA. After running the gas chromatograph test to see how much alcohol in the samples, I could conclude that after I received these graphs, that the new DNA in the bacteria had the same ethanol content as the bacteria did without the new DNA. And also, when compared to the 1% alcohol sample, there was no correlation, meaning that my new bacterial ethanol was not even close to 1% alcohol. I used my data and ran a statistical t-test from this, and I found that there was no statistical significance with my p-value between my bacterial ethanol produced with the new DNA versus the bacterial ethanol produced without the new DNA, meaning that they were the same and they weren't even close to 1% alcohol. So even though my hypothesis was incorrect, that I was not successfully able to produce a higher alcohol content with my new DNA, I was successful in the fact that I was able to successfully extract and transform DNA from E. coli into my Pediococcus bacteria. This was a great success for my project because this is a college level standard and I am a sophomore at Eastside. Because I was able to complete this process at Trine University, my first genetics part of my project was successful. And in the future, I would like to focus on the main genetics part of my project and not focus so much with ethanol production. So thank you.